So now I have the pleasure of introducing Dr. Hitoshi Kurumizaka, whose research focuses on the structural and biochemical features of eukaryotic transcription. Dr. Kurumizaka earned his PhD in 1995 from the Graduate School of Science and Engineering at Saitama University, where he studied DNA repair proteins. Later, he did a short postdoc with Professor Alan Wolf at the NIH in the US, where he focused his attention to the nucleosome. Most recently, he became a professor at the Institute of Quantitative Life Sciences, which is associated with the University of Tokyo. Throughout the last couple of decades, his lab has focused on reconstituting eukaryotic chromatin in vitro to generate beautiful structural images of nucleosomes and chromatin. Most recently, in 2018, both his lab and the Kramer Laboratory published the first images of RNA polymerase II transcribing through a nucleosome, forever amplifying insight to those of us interested in transcription and chromatin. Dr. Kurumizaka is not only a terrific scientist, but he's also an avid guitar player. So after this talk, I invite you to look for the Nucleosome song on YouTube to get a glimpse of his creativity. Thank you so much for joining us today, Dr. Kurumizaka, and please go ahead. Well, I think...
All right.
All right, got it.
Okay. Can you see it?
Yes.
All right. Okay. Thank you very much for giving me an opportunity to talk here today. Yeah, I'm very happy about it. Yeah, my name is Hitoshi Kurumizaka. Thank you very much for a great introduction for me. Yeah, today I will tell you about the dynamic structure changes in the nucleosome during gene regulation. All right. Okay. As everybody knows already, the genomic DNA is so huge in human. About 2 meters of genomic DNA is compacted inside of the nucleus. The nucleus diameter of nucleus is so tiny, about the 10 micrometer. To accommodate entire region of the genomic DNA into the nucleus, the genomic DNA must be compacted as a chromatin structure. Then basic architecture of the chromatin is the nucleosome core particle. The protein components of the nucleosome core particles are histones, core histones H2A, H2B, H3, H4. 2 of each core histones form histone octamer, and about 150 base pair DNA is wrapped around 2 times. In the left hand, we wrapped around the histone core. This is the basic architecture of the nucleosome core particle. And the chromatin. Linker DNA, nucleosome core particles are connected by the linker DNA and form the higher hierarchical architecture in chromatin. Then the DNA replication, transcription, recombination, and DNA repair must be promoted within the chromatin structure. It looks very difficult. Then the chromatin structure may be inhibitory for DNA action, this DNA action. However, the epigenetics use this system to regulate the genomic DNA function in genome. The epigenetics means heritable changes in gene expression that do not involve changes the DNA sequence. It may be responsible for chromatin structure. That is the best, one of the important questions to ask. Then today I'm going to tell you about 2 topics. First topic, how does chromatin dictate transcription initiation sites? Let's move on the first topic. Okay, first topic is related to the overlapping dinucleosome. And when you have a nucleosome around the transcription initiation site here, it is quite obstacle because the nucleosome formation generally inhibits the binding of the DNA binding protein to the genomic DNA region around the transcription initiation site, transcription start site. Then, as already Lucas told you, the chromatin remodeler may be recruited around the transcription start site and get rid of the nucleosome from the transcription start site. And they make the space and transcription machinery, including RNA polymerase II, recruited to the transcription start site. Then transcription is initiated. That's the basic, one of the basic mechanisms, how does chromatin structure dictate the transcription initiation site. Then the several laboratories already published a very, very beautiful structure of the chromatin remodelers complex with the nucleosome. I don't tell you much about it. The one of them is the CHD1 nucleosome complex. Lucas already showed you. It was the first one, if I'm correct, the first atomic resolution structure of the chromatin remodeler complex to the DNA. It is very nice work. Then today I'm going to talk about the chromatin structure after the chromatin remodeling may be promoted. Then just downstream of the transcription start site, after transcription, after chromatin remodeling may be promoted. Unusual nucleosome structure, probably the two nucleosomes hit together and form the unusual nucleosome structure. We call it overlapping dinucleosome, may be formed. Then two papers has been published by the Schnitzler lab and Owen Hughes lab. These papers are very nice, and they nicely shows that after the nucleosome remodeling, unusual dinucleosome structure may be formed. Then Tom's lab, Tom Owen Hughes lab, elegantly established the system to reconstitute the overlapping dinucleosome in vitro, and then they showed that in the overlapping dinucleosome, 4 copies of histone H3, H4, and 3 copies of histone H2A2B associated each other and formed the histone protein, and about 250 base pair, not the 150, 250 base pair DNA is wrapped around the histone core. This may be the overlapping dinucleosome structure. Then we just follow the method established by the Tom's lab. Then we reconstitute the unusual overlapping dinucleosome. Then first DNA substrate is just we employ the 601 positioning, the nucleosome positioning sequence. It is the great invention by the John Witham by the in vitro selection methodology. Then if you have a 601 sequence here, and then you assemble the nucleosome in vitro, you can have a 2 nucleosome with a linker DNA. This is the native page analysis. Yeah, this is the naked DNA. If you assemble the dinucleosome properly, you can get the band shift here. Then, if you get rid of the entire region of the linker DNA, you still have a dinucleosome nicely here. And then, if you truncate the 22 base pair downstream of the upstream 601 sequence, and then 22 base pair of the upstream region of the downstream 601 sequence, you can get the 250 base pair DNA with the 601 sequence repeats here. Then you can still have a dinucleosome kind of band here. It vibration little different from the prediction. Then, if you check the protein component contents of the unusual dinucleosome, you still see the histones, or core histones, H3, H2B, H2A, H4, nicely. Then we check the unusual dinucleosome is maybe the overlapping dinucleosome or not. Then to check it, I collaborated with Kazumi Saikusa and Satoko Akashi. They were in the Yokohama City University. They have a very excellent technique, ESI mass analysis, electro-spray ionization mass spectroscopy. In this method, actually, you can measure the molecular mass of the complex without disruption. Then, if the nucleosome, 2 nucleosomes just associate each other, and then you about the 250 base pair DNA is just wrapped around the 2 nucleosomes. The theoretical value of the molecular mass is 380 kilodalton. Okay, but if we reconstituted the overlapping dinucleosome correctly, it may lose the 1H2A to be dimer. The predicted the molecular mass is 350 kilodalton. Then this is the experimental value. Actually, we find out the reconstituted overlapping dinucleosome we have is the 350 kilodalton just. Then we go ahead to make the crystal and determine the crystal structure of the overlapping dinucleosome. This is the crystal structure of the overlapping dinucleosome we obtained. Then magenta part indicates the octasome part, and then cyan part indicates the hexasome part. In the overlapping dinucleosome, the histone hexamer and histone octamer associate each other and then form the core, histone core. Then 250 base pair DNA is continuously wrapped around the histone core 3 times without obvious DNA, linker DNA section. This is the structure of the overlapping dinucleosome. Then in this picture, the magenta indicates the H2A, H2B dimer location. This is the octasome part. When you look at this picture, you can see the 2 H2A, H2B dimer here, and then this is the hexasome part. In the hexasome part, one H2A, H2B dimer is missing at the from the interface between hexasome part and octasome part, and then you can see just one H2A, H2B dimer in the hexasome part. This is the structure. Then next question we had is that actually the overlapping dinucleosome is existing in the cell or not? This is a very important question. Then to investigate the overlapping dinucleosome existence in the cell, we need to get the probe. Okay, then we employ the MNase, micrococcal nucleus treatment assay. Then this is just regular dinucleosome. This is the overlapping dinucleosome. When you treat the regular dinucleosome by the micrococcal nucleus, MNase, MNase preferentially trim, digest the linker DNA region here, and the DNA directly wrapped around the histone core is resistant to the MNase digestion. Therefore, when you treat the dinucleosome with the MNase, you can get 150 base pair protection. This is the DNA region directly wrapped around the histone core. Then, if you repeat the same experiment with the overlapping dinucleosome, you can find that actually the entire region of the 250 base pair is resistant to the micrococcal nucleus attack. It indicates that the micrococcal nucleus did not digest the inside of the overlapping dinucleosome DNA wrapped around the overlapping dinucleosome. It gives you a nice probe to investigate the existence of the overlapping dinucleosome in cell. Then we prepare the chromatin from the HeLa cells and digested the chromatin by the micrococcal nucleus hardly until the mononucleosomal level, like this. Then we analyze the sequence by the next generation sequencing analysis, NGS analysis. We made a nice collaboration with the Okawa's laboratory, Kyushu University. They had the expertise of the NGS analysis of the chromatin. Then, Okawa. Then we prepare the HeLa cell chromatin and digested the HeLa cell chromatin until the mononucleosomal level here. You can only see the 150 base pair DNA segment. Then you can cut this band, and you can also cut this band about the 250 base pair DNA and then read the DNA sequence by the NGS analysis. Then map the DNA fragment onto the relative to the transcription start site. Then we repeated the experiment twice, number one, number two, independently. These are the results of the number one experiment and number two experiment. Then, first of all, when you look at this picture, this is the 155 base pair DNA segment plus minus 10 base pair. It means the nucleosomal signal. When you look at this picture, you can find that just downstream of transcription start site, you can see the first nucleosome, second, third, 4, 5. You can see the nucleosome alignment very nicely. Then, if you have overlapping dinucleosome just around the transcription start site. You can see the peak of the 250 base pair just around the transcription start site. This is the result. When you map the 250 base pair DNA fragment, you can see the clear peak just downstream of the transcription start site. It suggests that the transcription, the overlapping dinucleosome can be actually produced just downstream of the transcription start site. Then we repeated the experiment twice, and then the results are very consistent. Then we concluded that during the transcription initiation process, probably in the active promoter, nucleosome remodeling may be promoted around the transcription start site, and the just downstream of the transcription start site, the overlapping dinucleosome is hoped. Then next question is that the dynamics of the overlapping dinucleosome structure. Then to test it, we collaborated with the Kono Lab QST and Sugiyama Lab Kyoto University. Sugiyama Lab performed the SAX analysis, the solution structure analysis of the overlapping dinucleosome. Then Kono Lab performed the MD simulation analysis together with the structural information about the crystal structure and the solution structure. Then what we found is that in MD simulation analysis, interestingly, the histone H3 tail from the hexosome part bind to the DNA, nucleosomal DNA of the octosome part. On the other hand, histone H4 tail from the octosome part bind to the DNA of the hexosome part. These histone tails may bridge the two moiety of the hexosome part and octosome part. It restricted the dynamics of the overlapping dinucleosome structure. Then if the tail DNA contact may be lost, the dynamics of the overlapping dinucleosome is changed a lot. Probably the histone modification and the histone tail binding protein may be the nice regulator of the overlapping dinucleosome configuration and the overlapping dinucleosome dynamics. Then we want to see the structure variation of the overlapping dinucleosome. To investigate it, we perform the Cryo-EM analysis here. Then these are the very preliminary data in this point of time. Then please keep them in your mind only. Then we classified 12 overlapping dinucleosome structure by the Cryo-EM. Then this is the side views. Then the arrangement of the hexosome part and the octosome part may be different in these 12 classes of the overlapping Cryo-EM structures of the overlapping dinucleosome. Then this is the crystal structure of the overlapping dinucleosome. Then one of them nicely very close to the structure we determined by the crystal structure. Then the other structure is some a little bit different from. Then some of them are different. This one is very different. Even the angle of the hexosome part is totally different from the crystal structure. These are the dynamics of the overlapping dinucleosome. Then we concluded that an overlapping dinucleosome is kind of dynamic structure. It has a variation of the dynamics. There's such a variation may be regulated by the histone tail DNA binding. This is the interesting question to answer the future. All right. Then last year, I've been as you already know, I visited the Seattle. Then I found the headquarters Amazon already know the importance of the overlapping dinucleosome. They already built the overlapping dinucleosome here, like this. Now in this situation, everybody needs to get something from Amazon. Amazon already know the secret of life. Okay, in the overlapping the nucleosome subject, the Daiki did the crystal structure analysis, crystal structure of the overlapping the nucleosome analysis. Then Aki established the in vitro system. Then Hiroki Yoshi performed the cryo-EM analysis in this point of time. Then we have many nice collaborators I already introduced. Then the cryo-EM analysis, we collaborated with the Sekinei's lab, Haruhiko Ehara, took the data. Then let's move on the second topic. How does the transcription take place in chromatin? This is the fundamental question we already, actually Lucas already introduced you nicely. Then this is the picture of the transcription on the chromatin. It looks very obstacle. Even you have a nucleosome in front of the RNA polymerase II. RNA polymerase II must take care about the nucleosome core particle structure. Then in this point of time, we have no idea how RNA polymerase take care of the chromatin and nucleosomal DNA. Therefore, in this movie, the RNA polymerase just eat the nucleosome, just eating. Then to understand the mechanism, how the RNA polymerase II read the DNA sequence wrapped around the histone core, we established the transcription, in vitro transcription system. Then, first of all, we reconstitute the nucleosome with the DNA sticky end. The DNA with the sticky end here. Then, after purification of the nucleosome, this nucleosome, we ligated the bubble DNA here. Then, purify the nucleosome again. Bubble DNA is very important for a loading site of the RNA polymerase II. This region function as a nucleosome, the RNA polymerase II assembly site. Then this is the purified nucleosome. Then purified nucleosome containing histones H3, H2B, H2A, H4, 1212121 stoichiometry. In this case, we employed the histone H3.3 because 3.3 is dominantly assembled on the transcriptionally active gene. Then we purified RNA polymerase II from the yeast peak here. Then we also prepared the transcription elongation complex, TF2S here. Then we performed the transcription assay. We loaded on the RNA polymerase II onto the bubble region, linker region of the nucleosomal DNA, and then put the primer RNA primer here. Then determine the direction of the transcription orientation. Then transcription goes on. Then when RNA polymerase II hits the nucleosomal DNA somewhere around in the nucleosome, it will be stored. Then we purify the stored RNA polymerase II to the nucleosome by graphics and gel filtration method. Then go ahead to determine the structure by the cryo-EM analysis. That's the strategy. Then this is the transcription gel, the RNA gel. This is the primer RNA. Then in this case, we initiate the transcription from the -7 side. The nucleosome is a symmetric structure, as Caroline Luga already determined 20 years ago, more than 20 years ago. And then the center of the DNA is just sit on the axis of the nucleosome. It's called the superhelical location 0. Then every 10 base pair, the nucleosome DNA location is addressed -1, -2, -3, -4, -5, -6, -7. And then opposite side, nucleosomal DNA location is defined as a +1, +2, +3, +4, +5, +6, +7. Then totally about 150 base pair DNA is symmetrically wrapped around the histone core. That's the structure of the nucleosome. Then we put the bubble DNA here. Then transcription is from this side. Then we first find that RNA polymerase II stored a little bit around the -6 location here. Then stored a lot around the -5 position, as already Lucas introduced the structure of the -5 complex, maybe -6 complex. Then we probably the naming of the -5 complex, -6 complex is a little bit different of the definition of the Lucas structure. Probably Lucas structure is close to the -6 complex. Then when you look at this picture, you can also see the RNA polymerase II is storing around the -2 a little bit and the -1 a lot. Then in this construction, we designed the RNA polymerase II maybe stored just around the -1 position, but most of the stored RNA polymerase is just before the region we designed. It means that all stored position may be the naturally storing position onto the nucleosome. Then we performed the cryo-EM. We took the cryo-EM images, and the many states apparently exist. Of course, you can see the many states here too. Then when you look at this picture, you can see the nucleosome here and the RNA polymerase II here and the linker DNA here. Now it means that RNA polymerase II is not reached to the nucleosome yet. Then these guys are also the RNA polymerase II is still transcribing in front of the nucleosome. But when you look at these pictures, you can find that some of the RNA polymerase hit that interact with the nucleosome directly here. And then with this sample, we determine the snapshot structure of the transcribing RNA polymerase II nucleosome complexes, like this. Then we excited about it when we first got these structures. This is the -5. This is the top view, and this is the side view. Then this is RNA polymerase II stored just in front of the nucleosome. This is, we call this one is -6 complex. Then peel off 20 base pair of the DNA and store the -5 position alone. Then the RNA polymerase overcome this nucleosome barriers, peel off the 50 base pair more and store the -2 a little bit. Then 10 base pair, peel off the 10 base pair more and store the -1 position alone. Then this experiment shows that actually RNA polymerase II transcribing on the nucleosomal DNA with the gradual peeling of the DNA from the histone surface. Then this is the comparison of the Patrick Kramer's lab, Lucas Fanning and Cecil Boss published a nice, very nice structure. It quite close to the structure of the -6 complex of us. Okay. Then why the polymerase is storing around the -6 complex is one reason, major reason is the histone DNA contact. Here, at the edge of the nucleosome, the DNA is captured by the histone H3 arginine residues type 3. Then when RNA polymerase hit the DNA histone contact, it stored a little bit. Then after get rid of this histone DNA contact, then RNA polymerase peel off the 20 base pair of the DNA from the histone surface and hit second place. It is created by the interaction of the histone H2A arginine 77 with the DNA. This is very hard contact maybe because most of the RNA polymerase stored around the -5 position alone. Then after overcome the -5 barrier, the nucleosomal DNA is easily peeled off by the RNA polymerase II until the -2 position here. Then -2 in the -2 position, the RNA polymerase II hit the histone DNA contact again, and then get rid of this position and nucleosome barrier. Finally, RNA polymerase II stored at the just before the -1 position because of the histone DNA contacts are so hard around just before the -1 position here. Then another things we found is that in the complex of the -1 here, surprisingly, histone H2A2B is still existing. The previous studies have been shown that if you get rid of the entire region of the DNA around the histone H2A2B, histone H2A2B dimer is released from the nucleosome automatically, spontaneously, I mean. However, even the RNA polymerase II peel off the entire part of the nucleosomal DNA around the histone H2A2B dimer. It's still existing because of the direct interaction of the RNA polymerase II to the histone H2A2B in the complex of the superhelical location -1. This induced the DNA binding of the another nucleosomal link, nucleosomal molecule of the linker DNA of the nucleosomal DNA here. Yeah, we already got this structure. Then this may be the mimicking structure of the template looping. The mathematic loop elegantly shows that when the transcribing RNA polymerase hit the nucleosome, the upstream region of the DNA flip back and bind to the nucleosomal histones and form the template looping. Then upstream region of the DNA is disappearing because it is flexible. Then we already know that it doesn't. This DNA segment is not the upstream region of the transcribing RNA polymerase, but it may mimic the template looping structure. If the RNA polymerase is keeping the H2A2B dimer onto the nucleosome, that it can be formed. Then this is the images. Okay, we promoted the transcription reaction from the -7 side. We put the bubble DNA here. Then we initiate the transcription elongation from this side. Then RNA polymerase first hit the nucleosome without peeling of the DNA and stored a little bit. Then RNA polymerase II peel off the small part of the DNA, nucleosomal DNA, and stored around that -5 position alone. Then DNA as peel off more and stored around that -2 position a little bit. Then finally, RNAPII-2 stored around the -1 position alone. Then after passing through the nucleosomal dyad DNA, the entire part of the nucleosome may be disrupted. It has been shown by the by previous biochemical experiments alone. Then we also see the mimicking structure of the overlapping the template looping. All right. Okay, then now we know how the RNA polymerase II, RNAPII-2, transcribe the DNA wrapped around the histone core. Then Lucas already told you the nucleosome remodeler may function, like a CHD1, may function very importantly to get rid of the nucleosome barrier. Of course, histone chaperone like fact may be important for the activation of the transcription elongation. It is also very interesting fact may get rid of the remaining H2A2B in front of the RNA polymerase, the nucleosome located in front of the RNA polymerase II. It may enhance the transcription elongation. Of course, histone variants and histone modification change the histone DNA contacts within the nucleosome. It may affect the transcription efficiency. Then many things we need to do in future. Then today I'm going to tell you a little bit about the transcription elongation factor. Then famous transcription elongation factor, SPT45, it is also called the DSIF in mammalian cell. Then this is the excellent transcription activator. And then we tested the transcription reaction onto the nucleosome with SPT45 and another transcription elongation factor ELF1. Then this is the RNA gel. Then when you promoted the transcription reaction with the naked DNA, you can get the runoff product a lot. Then if you assemble the nucleosome, then the runoff product production of the runoff product is very decreased a lot. But instead of it, you can see the -5 stop and the -1 stop here. Then when you add the SPT45 here, you can see that great transcription enhancement. Yeah, you can see the more transcript about this until the -1 position, and then a little bit increase about the runoff product here. Then ELF1 alone did nothing. However, if you add the ELF1 and SPT45 simultaneously onto the nucleosomal DNA, these factors synergistically enhance the transcription elongation step onto the nucleosome. Actually, reduce the amount of -5 position and the increase amount of the -1 position and the runoff product alone. Then we have that when we found that synergistic action of the transcription elongation factor onto the nucleosomal DNA, we would like to know the structure of the transcription complex. Then we determined that -5 complex with the elongation factor SPT45L1. Previously, the Kramer lab and the Sekinez lab already reported these transcription elongation factor is stick on the RNA polymerase, the surface of the RNA polymerase 2 nicely, covered the surface of the RNA polymerase 2 nicely. Then it is located just interface between nucleosomal DNA and RNA polymerase. In Lucas talk, Lucas shows that the RNA polymerase 2 clamp head and some part of the RNA polymerase 2 directly interact with the nucleosomal DNA. It may be very inhibitory. Then transcription elongation factor inhibits the direct interaction between RNA polymerase 2 and the nucleosomal DNA here. It is the mechanism of why these elongation factor enhance the transcription elongation efficiency. Then if you compare the structure without elongation factor, actually RNA polymerase 2 directly bind to the nucleosomal DNA, as Lucas already showed. Our structure already already present the same result. Then if you add the transcription elongation complex, the AL1 just located just between the RNA polymerase 2 and the nucleosomal DNA, and move the nucleosomal DNA away from the RNA polymerase 2 and inhibits the direct interaction. It is the reason why the reduced amount of -5 stop and enhancing the transcription elongation through the -5. Then we also determined the structure of the -1 complex with the elongation complex. Then same thing, we saw that these elongation factors inhibit the direct interaction between nucleosomal DNA and RNA polymerase 2. It may be the reason why these factors enhance transcription elongation. Then we also determined two different kinds of -1 complex structure here. These two different kinds of structure has a two. One structure is progressive, one more basis. Then this movie shows that the one base progression of the nucleosomal DNA transcription like this. Yeah, 36-degree rotation of the nucleosomal DNA, nucleosome, you can find here. Then now we know the mechanism, how RNA polymerase II transcribe wrapped around the histone core, and how the transcription elongation factor reduce the nucleosome block -6, -5, -2, and maybe -1. Then these are the contributor. The Tomoya Yuka established everything about the in vitro transcription system. Then Haruhiko purified the RNA polymerase II. Then took the Cryo-EM images and determined the Cryo-EM structures a lot. Then together with the Tomoya. Then we intimately collaborate with the Sekine's lab, then Mikako's lab, then in Lincoln Institute. Then we are doing the transcription nucleosome experiment projects a lot. We are still doing many things together. Then finally, we wrote the song called "Go Around." Then in this song, I explained how to polymerase to transcribe on the wrapped around the nucleosomal DNA, nucleosome histone core. Please visit YouTube site and put the Kurumizaka here, and then see this icon. And then if you click here, you can listen the part 2 goes around new song. Then I already put the nucleosome song too. Please enjoy the nucleosome song and the part 2 song. Thank you very much for attention. I will take some questions.
Thank you very much, Doctor. That was fantastic. And of course, we invite everybody to go and check out the talks on YouTube so we can continue learning. So we have a couple of questions, but I just want to remind people, especially those that are joining us for the first time, that they can raise their hand and I can unmute them so they can go ahead and ask their question live for you. So while they do that, I'm just going to read some of the quick Q&A questions. The first one says, from Ashutosh Kumar, says, Professor Hitoshi, the last dinucleosome structure is very different, looks completely distorted. Can the dynamics change so much, or is this some other intermediate state?
Yeah, there are two possibilities. It is the intermediate state of the chromatin remodeling, or it is the permanent structure, one of the new fundamental structure architecture of the chromatin. We don't know the answer yet, but actually we know the stability of the overlapping dinucleosome. Stability of the overlapping dinucleosome is just same as the normal nucleosome. It may suggest that it is not the dynamic, very unstable intermediate. It is kind of stable structure, but very dynamic.
Okay, thank you. So we have a couple of people raising their hand. I think Jason Fan, do you want to ask a question?
Yeah. Yeah. Can you hear me? Oh. Okay. Yeah. Very, very cool talk. I was really interested in the your last experiment with the adding the factors and seeing that run on. I was curious, like, if you analyze the template basically after the run on has gone through. Do you think those say, like, if you do MNase or analyze by mass spec, do you think you would see like things like hexosome or shifted nucleosome position and things like that?
Yeah, that's very interesting idea. We are testing it right now. Okay. We didn't get a nice answer yet, but the system we used to use is that we truncated the opposite side of the DNA. Therefore, it is impossible to move the nucleosome away. Yeah. And then we put the linker DNA to the opposite side, and we are doing the transcription experiment and the structural analysis in this point of time. It may give you a nice answer after the transcription and transcription is promoted. What may be happening on the nucleosome structure? It may form the hexosome or whatever. It may form some other kind of structure. It may be very interesting.
Great, thank you.
All right, thank you, Jason. So Vivian Chen, she also has her hand raised. So I'm gonna unmute you, and whenever you're ready, go ahead and ask your question.
Okay, so I'm pretty impressed with all the work. Hello. So I have basically have two questions. So for the dinucleosome reconstruction, I'm kind of curious about like you said the H3 and H4 tails are like playing a role there, but have you checked the post translo modifications like profile like okay.
Not yet, but it is very interesting subject project we will do. Yeah,
okay.
And then of course we can also do the experiment with a tailless nucleosome. Yeah, we can prepare the truncation version of the histone and then make the overlapping the nucleosome, and we can see what is happening on the dynamics.
Okay, cool. So the next question is more like general question, but like during the reconstruction of the POL2 and the nucleosome complex, like how do you determine the DNA sequence or template you use for that?
Oh yeah, we tested the several sequences anyway. Then basically we use the 601 DNA sequence because we need to fix the position of the nucleosome anyway. Then I don't tell you details about the construction, but actually we put the first T just after the -1 position. Then we change all T residues, half of the nucleosomal region, because we can transcribe until the -1 position without T, right?
Okay.
And then we can store the RNA polymerase around the -1 position. That was the first strategy of us. Then we changed the ORT in front of the axis. Then we did the transcription experiment. Then recently we tested the transcription experiment from the opposite side of the 601 sequence, and then we also see the consistent result, -5 position stop and -1 position stop alone. It not depend on the DNA sequence.
Okay, cool. I see. Thank you so much.
Yep.
Thank you, Vivian. So Mustafi, Pallavi, so she also has her hand raised. So whenever you're ready, go ahead and ask your question.
Hello. Yeah.
Hello.
Amazing talk. I was wondering, so it's kind of similar to the last question asked. So the overlapping dinucleosome, the dynamics will be different in different genomic regions, and will the post-translational modification of H3, H4 determines the dynamics of the overlapping dinucleosomes in the different genomic regions? Do you think so?
That was the idea we have right now.
Okay. And one more question was that in the elongation factors that you see SPT4 and 5, so there are the other kinases which also interact with SPT4 and 5 during the elongation process. Do you think if those factors also like kind of enables this process of not letting direct binding between RNA POL2 and nucleosome DNA, and thus by letting the elongation continue? Do you think
That's an interesting idea. Yeah. Maybe it is testable as long as we can reconstitute the in vitro transcription assay. Yeah. Is that correct answer for you?
Yeah, that's Pallavi. Is that? Do you have any other question?
Oh no, thank you.
Okay. All right. Well, I think that's it.
Anyway, Melvin, thank you very much for you writing the very, very, very fantastic review, the introduction of us. I was so happy to see it.
Thank you. It was a lot of fun.
Thank you very
much. John and Ron Conaway. We
really lion picture. I like
that. Thank you so much for joining us today. Thank you so much, Lucas, and everybody else who was able to join us for this special time seminar of Fragile Nucleosome. We really thank you, and we really appreciate that you're here. In the future, we're going to go back to the normal times, but we invite you to either join us on YouTube, watch the talks, or find us here in the Discord channel so you can continue communicating with us. And everybody else who woke up super early for this, thanks so much for this.
Thank you.
Thank you. Have a good one.